So in continuation with our previous lectures, we discussed about replication rule of 3, attachment, penetration, uncoating, then central dogma part, and today we'll discuss assembly, maturation and release of virus from the cell. This is the upper respiratory tract. We'll take example from this portion with respect to influenza viruses. So the first is the assembly, where this assembly takes place. So for different viruses, sites are different. Number one, so for different viruses, these assembly sites are different. For example, number one, if we start from the nucleus, number two, this Golgi, then number three, cytoplasm, and number four, plasma membrane. So these are the different sites where different viruses assemble. So in case of nucleus, Golgi, cytoplasm and plasma membrane. So for different viruses, we have these different sites. For example, in case of nucleus, adenovirus and herpes. These viruses, DNA viruses, they assemble in the nucleus. Then inside the Golgi, there is one called Bunia virus, which assemble inside the Golgi body. Then proper cytoplasm, picoRNA or reoviruses, they assemble in the cytoplasm. Then plasma membrane. Plasma membrane is your orthomyxovirus, that is influenza virus, paramyxovirus, then rhabdoviruses, that is rabies viruses, then retroviruses and toga viruses. So these viruses, they assemble at plasma membrane and cytoplasm, both places. So these are the different viruses and these are the different sites of assembly. These viruses you can remember with the mnemonic parrot, PARROT. You can remember with parrot. Parrot means, PA means paramyxo, R means rhabdo, next R means retrovirus, and O means orthomyxo, T means toga viruses. So these parrot viruses, they assemble at plasma membrane and cytoplasm. So from these different viruses and their assembly site, we'll discuss orthomyxo as an example. As we know, the orthomyxovirus, they affect in human upper respiratory tract, and in case of birds, for example ducks, it affect the gut epithelium. So this is the upper respiratory tract of human. Similarly, this is the gut of avian gut. So these are the cells. If we take one cell from this portion, this is the respiratory cell, and this cell contains some receptors which are known as alpha 2,3 or alpha 2,6 sialic acid receptors. So these are the receptors present over the respiratory epithelial cell. Again, the virus is having handshaking domain that is known as heme agglutinin, HA, heme agglutinin. So this is the interaction. HA, this HA is having two parts. This is having two parts. This is like, this head is known as HA1 and this tail is known as, this is HA1, this is HA2. This one portion interact with the sialic acid receptor. So here is the attachment, then penetration, and here comes the, this is the endosome, inside it, this is the virus. This portion, HA2 portion, this HA2 portion helps as a, this is known as HA2 portion. This is known as fusion peptide. Fusion peptide. So in case of orthomyxoviruses, this HA portion is having two parts, HA1, HA2. This one portion interact with the sialic acid receptor, and this HA2 portion, it acts as a fusion peptide. Where the fusion step takes place, the viral envelope fuse with the endosome membrane. And what is the result of this fusion? These 8 segments, it comes out. This is known as uncoating. Uncoating. So the genome is coming out of this. So the first role of HA1 is the binding with this receptor, HA2 for fusion peptide. But in case of paramyxoviruses, these both functions, they are carried out with different proteins. They have fusion protein on their surface, like which is known as F. In case of paramyxo, they have H protein for attachment and they have F protein here for the fusion. But in case of orthomyxo or influenza viruses, the same glycoprotein HA act as attachment, also act as a fusion peptide. So this is the difference between ortho and paramyxo. So this genome will enter the nucleus. This genome will enter the nucleus. So this is the genome. It enter the nucleus. Then this genome, which is negative sense. This is negative sense. It uses RDRP enzyme and it is converted into plus polarity messenger RNA. Plus polarity messenger RNA. This messenger RNA comes out in the cytoplasm where you have free ribosomes. Here synthesis of early proteins. And these early proteins are M1 protein, nucleoprotein, and polymerases. So some free ribosomes are in the cytoplasm. They've synthesized these early proteins. These early proteins, they enter and they help in the replication. So suppose these are replicated genomes. So after replication, this portion will form, this is the endoplasmic reticulum. On this endoplasmic reticulum, protein synthesis takes place. Protein synthesis take place. So which proteins are synthesized here? Structural proteins. Structural proteins. Heme agglutinin, neuraminidase, and M2 ion channel. So these are the surface glycoprotein of influenza virus. They are synthesized here on the endoplasmic reticulum. Then endoplasmic reticulum is in connection with the Golgi body. So these three proteins, they are packed inside the vesicles and they are released to the surface. This point. So these proteins, they anchor themselves over the plasma membrane at some places. For example, at this place also. So these are HA, NA, M2. These are the surface glycoprotein of influenza viruses. Then some these they enter inside this. These are the genome and these are the structural or non-structural, whatever. They combine and what they are forming. This is the 8 segment genome and outside is the M1 protein. This is the M1 protein. M1 protein assemble here. So this is known as viral ribonucleoprotein complex, where nucleoprotein also assembled and M1 protein. So this complex will go near to the plasma membrane. At this point, on the plasma membrane, these surface glycoproteins, they are anchored, and this portion will come here and it will cover as an envelope. So this is the assembly or formation of the complete virion with all the surface envelope glycoprotein. Then after this assembly, we'll discuss maturation and release. So these are the steps where the viral ribonucleoprotein complex, it moves under those part of the plasma membrane where surface glycoproteins, which are synthesized in the endoplasmic reticulum, they are anchored at these places. Then there is fusion of this as in the form of an envelope, and this portion will come out of the cell. This will come out of the cell. So after assembly, our next step is the maturation step. Our next step is the maturation. Maturation means this virus particle is having the surface glycoprotein Na and M2HA. This particle is immature. This is non-infectious. This is non-infectious. So some structural changes will take place. Some structural changes will take place and it will convert it into infectious one. It will become mature. It will become infectious. And this maturation step may take place inside or outside the cell. Outside the cell means in the respiratory mucus, in the respiratory mucus, in the lumen. So some strain of influenza viruses, they mature inside the cell, inside the Golgi body, where one enzyme is known as furin, which help in the maturation. So what is the meaning of maturation here? This HA, this protein is synthesized as a precursor protein HA0. So this furin or extracellular proteases, what they do is they convert this 0 into HA1 plus HA2. So now it is functional. It means this part is known as HA1 and this tail part is known as HA2. So this particle, it is having both the part HA1 and HA2. So why it is infectious? Because of this cleavage, this portion is able to attach itself with the salic acid receptor. And once it attaches here, after endocytosis, this HA2 will help in the fusion and uncoating. So if you take this virus and you try to infect a healthy cell in this part which is having HA0, this virus particle is of no use because it is immature, it is non-infectious. So this cleavage, this maturation may take place inside the cell in the Golgi body with the enzyme furin, or it may take place inside the lumen of the trachea, where mucus is there and some protease enzymes are there in the lumen. So they cleave this portion. So they cleave this HA0 into 1 and 2. 1 for the attachment with the salic acid receptor and 2 act as a fusion peptide. It will help in uncoating. So now this is mature and infectious. So what is the need of maturation? This step will convert inactive non-infectious virion into a complete infectious one. Similarly, if we look at flavivirus, yellow fever virus, Zika virus and all flaviviruses, they also undergo cleavage inside the Golgi apparatus and the enzyme is the same, it is the furin. So there are some protein over here and what it convert? It convert pre-ME complex into pre-membrane protein plus envelope protein. So this cleavage is important for the virus to become mature, to become infectious. So maturation may occur inside the cell or it may occur outside the cell. So in flavivirus, it occurs inside the cell, Golgi cell organelle with the enzyme furin, where this complex is cleaved and it will become active. So the last step is the release. So if we look at the release, As we know, most non-enveloped, that is naked virus, and number 2, most enveloped virus. Their release mechanism is known as cell lysis and their mechanism is known as budding. Their mechanism is known as budding. Budding. So, if this is the cell, some viruses they bud from the apical surface and some from the basal surface. Some viruses RIP, rest in peace. Why they are rest in peace? They bud from the apical surface of the cell. And what are these RIP? Rabies, influenza virus, and paramyxo. So these three they comes from the bud out from the apical surface. Then from the basal surface we have alpha virus, we have lentivirus, and one more is the vesicular stomatitis virus. So alpha virus belongs to family Togaviridae and lenti belongs to retroviruses. So some virus they bud out from apical surface, some from the basal surface. So from the apical surface they are the RIP, rabies, influenza, paramyxo, and from the basal surface alpha, lenti, and vesicular stomatitis virus. So budding sites, different viruses have different budding sites. And viruses example. This is the nucleus. Endoplasmic reticulum. And this is the Golgi with its vesicles. Golgi. And this is the plasma membrane. So we'll start from the plasma membrane. So there are some viruses which bud out from the plasma membrane. From the plasma membrane, they are again the RIP viruses, that is rabies, orthomyxo, that is influenza, and paramyxo. So they bud from the plasma membrane. Number 2 is from the Golgi, from this Golgi. Pox virus and Bunia virus. They bud from the Golgi apparatus. Then endoplasmic reticulum, coronavirus and flavivirus and flavi means yellow fever virus. Then nucleus, at the end nucleus. From the nuclear membrane, nuclear membrane they bud out. And there is one example, herpes viridae. So these are the different budding sites for different viruses. Plasma membrane, Golgi, endoplasmic nucleus. These are the examples. Then role of some enzymes in case of release. This is the influenza virus with 8 segment. It is having this hemagglutinin and this one is neuraminidase. This hemagglutinin, this is known as H1 and this is known as HA2. This role of this is attached with salic acid receptors. Salic acid receptors in the respiratory epithelium or gut epithelium. Then what is the role of this Na? As we discussed, HANA and this one M2, and this is the complete number 8 segment. Then nucleus. The role of this nucleus is to exit from the cell, exit or release from the cell. What happened if we block this one? What happened if this neuraminidase is not working, is not functional, or less in number? Then this virus is having HA. Again, this virus with HA will attach the same cell and it will enter, and again it will cause the infection to the same cell. So what this Na is doing, this Na is destroying it is destroying or it is cutting the linkage. For example, this is the salic acid and this is the galactose subunit. It is cutting this alpha 2,3 or 2,6 linkage so that virus will go out of the cell. Otherwise, the virus will clump or it will again cause infection to the same cell. So the main role of is Na is the release from the cell by cleavage of salic acid receptors. Otherwise, it will again attach to the and cause infection, or these viruses will clump over the cell membrane. So this was all about the replication of viruses, and this replication was productive because in this replication we discuss all step: attachment step, penetration, uncoating, then it may enter the nucleus, then multiplication, it will come out, then form the messenger RNA, or then some enzymes like RDRP, they will enter, they will cause the replication. Some of will cause mRNA, then again protein synthesis, this will enter, protein will, these are the structural proteins, they come out, these are structural protein, then these two combine, that is assemble, then here is the endoplasmic reticulum, here is the Golgi. After assembly, here they go maturation, then release out of the cell. In this way, this virus enter and exit. This is known as productive replication. In the next lecture, we'll discuss defective replication, not productive, defective replication.